Welcome to the Basics of Animal Development module. And in this talk, we're going to look at the role of developmental biology in evolution, as well as have an introduction on medical embryology. In this talk, we learn about how developmental biology helps us in understanding evolution, as well as an introduction into developmental defects. Developmental biology can help us understand the process of evolution. Now, based on Carl Ernst von Bayer's laws of development, Charles Darwin put forth the idea that the community of embryonic structure reveals a community of descent. Now, let us see what he's trying to say in that statement. So, if we take an example of an organism called as a barnacle and look at its structure, so from this picture, we can see that a barnacle seems to have a shell-like structure, and so it might be similar to snails, because snails also have shells. Now, another creature that lives in the ocean are shrimp. Now, when we look at the body structure of a shrimp and compare that with the barnacle, they look very different from one another. Now let's look at the embryo. So here is a picture of the embryo of a barnacle, and let us look at an embryo of a shrimp. Now when we compare these two embryos together, the embryos actually look very similar. Now the adult barnacle resembles a snail, and snail belongs to the group of mollusca, while shrimps belong to the group of arthropods. However, when we look at the embryonic structures, because of their similarity, it would make us group them into the same group, which is arthropods. Now, it turns out, by doing sequencing studies, we've actually found out that barnacles are more closer to shrimp than to snails, and hence they actually do belong to the group of arthropods. Thus, the relationship between groups can be established by finding common embryonic structures. And thus, a community of embryonic structures can reveal a community of descent, which in this case states that the barnacle and the shrimp, they belong to the same group because their embryonic structures have common features. Another way developmental biology helps us understand evolution is by the presence of homologous structures, which are structures that can have different appearances and functions but are derived from the same body part in an ancestor. In the picture, we're able to see the forearm of a human, a seal, a bird, and a bat. And in all of these organisms, the forearm has different functions. However, when we look at the arrangement of the bones, all of them have a humerus that is linked to a radius and an ulna, followed by the phalanges, which form the different appendages of the fingers. Now, we're able to see all of these bones in all of these animals, even though these structures have different appearances and functions. And this is because during embryonic development, they all follow the same pattern of development, which is the formation of the limbs. Thus, by studying the development of these four organisms, we can see similarities in limb formation indicating that they have arisen from a common ancestor. Thus, homologous structures are evidence for evolution. Homologous structures are different from analogous structures because analogous structures are structures that perform a similar function but do not derive from a common ancestor. So the example for this is the wings of the butterfly and wings of birds, and both of them are used for flying. However, when we look at the embryonic development of these two organisms, there is no common feature that gives rise to these wings, and hence they are analogous structures. Development is a very complex process that requires many different things to happen at the right time. And hence, there are defects that can happen during these developmental processes that result in birth defects. In the case of humans, approximately 2 to 5% of infants are born with visible anatomical abnormalities, which usually arise due to developmental defects. Now, abnormalities can be caused either due to genetic defects or due to exogenous factors or factors that are in the environment. Let's look at some terms involved, and one term is malformation, which is a developmental abnormality that is due to a genetic defect. This genetic defect could arise due to a mutation in the genes or due to the inheritance of genes that are defective. Another term is a syndrome, which is a condition where two or more malformations are expressed simultaneously. When you have developmental abnormalities that are caused by exogenous factors, they're called as disruptions. And an agent that is responsible for disruptions is called as a teratogen. Now, the field of teratology studies how teratogens disrupt normal development. One of the most well-studied teratogens is thalidomide. Thalidomide was a drug that was given to pregnant women to treat nausea that normally occurs during the initial stages of pregnancy. However, it was found that when women were given this specific drug, the drug actually caused birth defects. Scientists that started studying thalidomide found that a single dose of thalidomide was actually enough to cause birth defects. Now, one of the most common birth defects is shown in the figure, where we are able to see that there are problems with limb development, and thus the limbs that are formed are called as flipper limbs. Now, scientists were also able to determine what kind of defects would happen depending on when thalidomide was taken by pregnant women. And that is what is shown in the figure to the right, where we're able to see the days on the x-axis and the kind of defects on the y-axis. Thus, thalidomide has been considered to be one of the golden cases of a well-studied teratogen. With this, we come to the end of our talk, where we learned about how developmental biology helps us understand evolution and how developmental defects can be due to the defects in genes or due to exogenous factors.